Studies of the brain carried out by means of neuroimaging and electrophysiology show that the brain's motor circuits are activated when we perform actions. What is surprising, however, is that it has also been shown that they are activated when we observe actions performed by others and when we hear actions described. The research group, led by Professor Manuel de Vega at the University of La Laguna, has a long and internationally recognized track record in this field. Their current research uses cerebral neuroimaging and electrophysiology to study the neuroanatomy of how we understand language referring to actions.
It's important to make two things clear about human language. Firstly, it's a unique phenomenon in nature. There are no other animal species with language like ours, which combines thousands of words through which we construct and understand new phrases constantly. It's because language is creative that we are able to communicate with one another. The second important point is that language is a product of the brain. Language is a function of the brain, and this means that if we are to understand the machinery, the physical mechanisms, we have to turn to neuroscience, that is, to the study of the brain and of how the brain operates when we understand and produce language. Our main aim was to come to understand how the brain processes the meaning of language. In fact, a good deal has been known about the neurophysiology of language for quite a long time. For example, in the 19th century, it was known that there were two structures of the brain in the left hemisphere that are responsible for language. Proof of this is the fact that if there is brain damage as a result of an accident or a stroke or whatever, the person will tend to have a language disorder such as aphasia. We already know this, but we are taking the research a step further because we want to understand linguistic meaning, how linguistic meaning is processed in the brain, and what we have chosen to research specifically is language referring to actions. Why language referring to actions, you might ask? Because much of what we say throughout a typical day actually has to do with actions. We describe movements, for example, actions of the hand like catch, grasp, throw, press, cut, or actions of the body like jump, dance, run. So we talk about actions a lot of the time.
It has been observed that the brain is not only activated when the person performs actions, but that motor areas are activated when actions are observed or when we listen to sentences describing actions.
For example, when I hear the sentence, Luis cuts the bread or Luis throws the ball, areas connected to hand movements are activated, whereas when I hear Luis jumps the puddle or Luis dances, areas related to the movement of the feet and legs are activated. This is really quite extraordinary because we're no longer talking about the classic areas of the left hemisphere, but about other areas of the brain that have to do with motor activity. This is what we're looking into through a variety of techniques.
Neuroimaging techniques are used to obtain and observe the results. Magnetic resonance equipment is used to register changes in blood flow to the various parts of the brain. This flow shows that these areas are involved in a specific task.
When people watch videos showing manual actions or read phrases that describe manual actions, the very same or similar motor areas are activated as those that are activated when they actually carry out the actions. The functional aspect of the technique involves not only registering an image but also the metabolic changes and the changes in blood flow to specific zones. These changes in blood flow are what provide the functional image. That is, they show that a certain task activates a specific motor region which is not activated when another kind of task is involved. So in our case, a language action task such as cut bread activates a particular motor area, but a phrase that refers to another kind of action, for example, listen to music, doesn't activate that motor area. So what we are seeing through high-resolution imaging techniques is which areas are activated and which are not.
By means of cerebral electrophysiology, it is possible to register the brain's electrical activity. Here what we have is in fact a second measure, brain dynamics, which is used by researchers to better understand the way the brain works.
When we watch action videos or hear phrases such as cut bread, we get the same type of brain modulation, the same change in brain rhythm as when we actually perform the action. So it all comes together, both the images obtained and the records of bioelectrical changes in the brain provide the same information. Through this technique and through rather complicated statistical analysis, it is possible to detect changes in brain rhythm which occur as a result of motor processing. When you perform a motor action, a kind of signature is produced which indicates brain rhythm. This signature appears when we carry out an action, but also when we observe actions or understand language referring to these actions.
Another more modern technique is transcranial magnetic or electric stimulation. This technique involves stimulating or modifying brain activity in a particular zone for a few moments. By means of this technique, it is possible to excite areas of the brain or to inhibit their activity and then to observe the consequences of this.
What we have seen is that when we inhibit the motor areas of the brain, understanding of language referring to actions is negatively affected. This is an interesting finding in relation to these motor areas.
Researchers at the Brain and Discal Studies Laboratory work with cerebral patients, especially those with diseases like Parkinson's disease. These patients suffer from impaired mobility, and although medical treatment exists for the disease, Professor Manuel de Vega's group has observed that, aside from deterioration in mobility, there's also a reduced ability to understand actions they observe and language about actions.
In the case of brain patients, especially those with mobility disorders, we are aiming to do something very important, namely identifying early markers of Parkinson's disease. What does this mean? Currently, Parkinson's and Alzheimer's are diagnosed very late, by which time the disease has already been developing for many years. If we were able to find an early indicator several years before the disease begins to show itself, it would be a major advance because treatment could start earlier. There could be rehabilitation or other methods of slowing down the development of the disease. We believe that this is what we have found by detecting that Parkinson's sufferers show alterations in referring to actions and physiological markers that can be observed by electrophysiological brain records. This could allow for the development of an early marker in the case of suspected Parkinson's. We would therefore be able to detect the disease well before other tests. It would be an indicator based on recording of rhythmic electrophysiological activity in the brain, which we know through the various experimental studies we've conducted to be impaired in patients with Parkinson's.
With the aim of developing useful synergies and collaborations, the Brain and Discal Studies Laboratory works with other research centres, universities, hospitals and private companies.
We have a long history of international collaboration and have also worked with other Spanish universities. It's certainly not the case that the only outstanding work is done abroad. There are plenty of excellent Spanish researchers. We have very strong links with the University of Oviedo, the University of Granada and the Complutense in Madrid. We also have international links going back many years with the University of Wisconsin, the University of Oregon and the University of Memphis. More recently, we've begun to concentrate on Europe and are now working with the University of York in the UK, Freiburg in Germany and the Max Planck Institute in Holland. The ideal future is very simple. We'd like to be able to go on doing what we're doing and make connections with the real world, in hospitals where these patients are treated and with businesses capable of developing the appropriate software for the detection of the early markers, for example. I mean, computer businesses and others who might collaborate in the development of second language learning methods. What is very clear to us is that the basic research must continue. Before anything else, we need to understand the brain mechanics of language, but we also need to be clear about making sure that what we learn reaches the society at large. We need to make even more connections with the business sector, though we do already have some such links. In our group, the training of new researchers has always been a high priority because the greatest contribution we can make here in Spain is through the training of competent researchers.